All right, so thanks again for coming here. We'll introduce the first speaker, Professor Jian Tang. So Professor Jian Tang is currently an associate professor at Mila, the Quebec Institute of Machine Learning and AI, and holds a Canadian Cypher AI chair. He's also the founder of Biogeometry, a new startup in the field of AI for protein design. His main research interests are in deep generative model, graph machine learning, and their application to drug discovery. He's an international leader in graph machine learning, and his representative work, line on node representation learning, has been cited more than 6,000 times. His group has also pioneered some work on AI for drug discovery, including the first open source machine learning framework for drug discovery, Torch Drug and Torch Proteins, that are widely used. He was also recipient of Best Paper Award at ICML and Best Paper nomination at Triple W. Thanks a lot for coming.
So first, I would like to thank Dominic and the team for organizing such a great event. And it's very exciting to see the community is growing at a very fast speed. And welcome all of you to Mila. Okay, so in today's talk, I'm going to talk about what we have done in the past, like pretty much like 3 to 4 years on geometric deep learning for proteins. So I will start with the first slide to basically to tell you, okay, why protein is very important. Why do we need to work on proteins? So like in living cells, most of the work are actually done by proteins, right? So you have DNA translate to RNA, DNA. So you have a DNA should be transcript to RNA and RNA translate to proteins, right? So most of the work should be done by proteins. That's why in reality, it's very important to understand the functions of proteins, which have applications in a bunch of like domains. Let's say therapeutics, right? You have antibody drugs, or in industrial applications, you have like enzymes, or you have like materials. Okay. And then so nowadays actually we got a huge opportunities for machine learning models to understand the functions of proteins, because now we got a huge amount of data sets, right? For example, for sequence data set, so we have a huge amount of like a sequence database in UniProt. So we got like I think like tens of billion protein sequence available. So that's one type of data set which is really sequence, right? And the second type of like data is really structured. So we got a lot of like PDB crystal structures in PDB, which we have like maybe around like 300,000 structures, which help us to understand protein at the atomic level, right? And this is the second type of data we can leverage to understand proteins. And the third type of data is really like experiment data. Like nowadays we got like a really high throughput assays, so we can test like hundreds or thousands of proteins at a single bioassays, and then we can get the functions of each protein, right? So all this kind of data actually help us to better understand the functions protein. Okay, so in the past few years, there's a lot of work in the machine learning community for protein modeling. Of course, the easiest approach is sequence modeling, right? So you represent proteins as amino acid sequence, and then you build protein language models, right? You just train transformers BERT on top of those unlabeled protein sequence, and then you fine tune those transformers based on some limited labeled data, right? So that's pretty much the main I think the most popular approach for protein modeling in the past few years. But then later we realized that okay, there's a better alternative, because like for proteins, the structures determines the functions, right? And that's why in reality, so if we can better, if we can model proteins, if we can represent proteins as structures and then learn repetitions based on the structures, and that's a better approach. And that's why now people started to move from sequence modeling to structure modeling. But how do we model structures with machine learning approaches? That's we need to use geometric deep learning approaches. And that's basically the focus of this talk. Okay. So essentially we have three kinds of like research questions to answer for structure modeling. The first question is, okay, suppose now we have a 3D protein structure. How do we represent a 3D protein structure, right? Because like in deep learning, we pretty much map everything to a continuous vector, right? We map a word, a sentence, an image to a vector. But here now we have a 3D protein structure. How do we map a 3D protein structure as a continuous repetition, right? And this is the first type of like research question we have to answer. The second one is like in reality, because it's very difficult to obtain the 3D structures of molecules or proteins, right? Because we need to do like, let's say, crystallography, which is very time consuming and expensive, right? But then that's why we really need to rely on computation methods like AlphaFold2 or AlphaFold3 to predict the 3D structures of proteins or their interactions. And that's the second type of like a fundamental problem we need to tackle. Okay. And of course, at the end of the day, the most important problem is, okay, we always want to do generation, right? Generate models, we want to generate new proteins with good functions. Okay, we want to build diffusion models or flow matching models to design to generate new proteins. So that's basically the three fundamental questions we have been working on in my group in the past few years. So I'm gonna start with the first type of like research question. Okay, so suppose we have 3D protein structures. How do we learn the repetitions? So this is a work we published in last year's iClear. So essentially, we built a geometric encoder to encode the 3D protein structures. So the input here is like, so we have like a 3D protein structure, okay? And then what we do here, given this 3D protein structure, we first construct a graph. So here is a graph. So this is a graph of residue. So every node here is a residue, okay? Of course, then the problem is, how do we define the edges between the residues here? So we basically mainly make use of two kinds of information. The first type of information is of course the sequence information, right? Because protein is still a sequence amino acids. So that's one type of information we can leverage to build the edges between the residues. The second one, of course, is really the 3D structures, right? So here, what we do is essentially we just construct k-nearest neighbor graph. So for every residue, we just connect this residue to the most the closest residues, and that's how we got the graph. Okay, of course this graph is like not undirected; it's a directed graph, right? And then the problem of learning this protein structure repetition translate to learning the repetition of this graph, right? And then, of course, we know how to learn the repetition of graph. We just use graph neural networks, right? So we do massive parsing between the nodes or between the residues. Okay. Of course, when we do massive parsing, because here we have multiple different type of edges between the nodes. So when we do massive parsing, we have to take the edge type into consideration. But anyway, so we're gonna do massive parsing between the nodes, between the residues, okay? And then once we do massive parsing, so every residue, every node has a repetition, and then we can do pooling, right? So we pull all the residue repetitions, and then we can get the repetition of the whole graph, and that's the repetition of the whole protein. So this is the first type of methods we developed, which we call a GearNet. So for the GearNet, essentially we are doing node level massive parsing because we pass messages between the nodes, right? But then we actually come up a better solution, which we call like edge level massive parsing. So in the second type approach, we are not doing massive parsings between the nodes. So we are actually doing massive parsing between the edges. So why do we do this? Because remember that here, this graph is not a 2D proportion graph like what we usually see. So this graph is a 3D graph, right? So there's some geometric information we haven't leveraged in this graph. For example, so the edges, we actually have angles or torsion angles between the edges, which we didn't make use in the first model. And that's why in the second model, we do massive parsing between the edges. And then when we try to do massive parsing between the edges, we actually make use of the angle information between the edges. Let me give you one example. Let's say here, the central edge we are considering here is like 4 and 7, okay? And then, so we're gonna do massive parsing between this edge, 4 and 7, between all the edges who share the ending node with the starting node of this edge. You see like, so the central edge is 4 and 7. All the neighboring edges are 2, 4, 6, 4, whose ending node is the same as the starting node of this edge, right? And when we try to do massive parsing between the two edges, as I said, we take the angle information between the two edges into consideration, and that's why we're able to better make use of the 3D geometric information in the structure. Okay, so once we do the massive parsing, so then every edge has a repetition, and then we further do pooling. So we pull all the edge repetition, and then we can get the repetition of the whole graph. Okay, so that's what we call like GearNet edge, which is essentially doing like edge level massive parsing. Okay, and then for all these models, we're gonna train this encoder based on supervised data. Okay, so this is the supervised training part. And as we know, like in biology, one challenge problem is always we do not have so many label data, right? And that's why we always want to pre-train it. Okay, and that's why we come up with the second part. We're gonna pre-train this like the geometric encoder I just mentioned. So we're gonna pre-train this 3D geometric encoder based on a huge amount of like unlabeled structures, for example, the PDB structures, right? So which is really a lot, or the recent like release like predictive structure by AlphaFold 2 or AlphaFold 3. Okay. So here for the pre-training, we actually use an idea called like contrast learning, and so this idea is very similar to what we have seen in image pre-training, right? So you know like for image pre-training, they use contrast learning. So idea is very simple. So if two patches are from the same image, then their repetition should be similar. Otherwise, their repetition should be dissimilar, right? So here we actually take a similar approach. Of course, here we have to define what does patch mean on proteins on a 3D protein structure, right? So here we actually don't use a patch. We actually use like a motif or structure motif. So here you can see, okay, given this 3D protein structure, we can extract two motifs from this protein structure. Okay, of course, actually there could be two different ways to extract a substructure or motif from this protein. So one way is just like you just take a subsequence. You just take a subsequence from the entire sequence, okay, and then you can get a subgraph, and that's one way to get the motif based on the sequence information, right? So another way is, okay, you really like get the substructure based on the spatial information. So what we do here is, okay, we just randomly sample residue on the graph, okay, and then you specify a radius, and then all the radius lie within the radius will be taken into this subgraph or into this like motif, okay. And then for these two motifs, if they are from the same protein, so their repetition should be similar. Otherwise, their repetition should be dissimilar. That's essentially the idea of like contrast learning. And then we pre-train the model based on a lot of like unlabeled protein structures, and that's the pre-training part. Okay. And then in terms of like evaluation, so we evaluate our models on a bunch of like important like protein understanding tasks, which are quite all these tasks are quite like popular and classic. For example, enzyme commission number prediction, gene ontology term prediction for the classification and reaction classification. So these are the supervised tasks to evaluate the performance of our models. And that's we also have a pre-training data set. So one way to pre-train is really like we pre-train on the PDB structures, which are experimental structures, which are more accurate. But you can also pre-train the models on the predicted structure by AlphaFold or AlphaFold 2. Okay, so that's the pre-training part. So we can take a look at some of the results. So we have two kinds of evaluations. So in the first evaluation, it's pretty much like supervised learning. So we want to evaluate the performance of different encoders. So the first type of method is essentially like sequence-based methods, because you know, like we represent proteins as a sequence. So we have different sequence encoders like traditional CNN, LCM, or transformer, right? And then we can also represent proteins as graphs, okay? So which is the second type of like methods. And then the third type of methods is a method we propose, which we represent proteins as 3D structures, and we train a 3D geometric encoder. So in general, you can see that like for the supervised learning part, of course, our methods can get better performance because we actually make use of the entire 3D structure information compared to the sequence-based and also the graph-based methods, which is not very surprising, right? For the supervised training part, okay. And then you also see even on the if you compare the performance between GeoNet, which is doing massive passing between the nodes, and also GeoNet Edge, which is doing massive passing between the edges, you can see that. So GeoNet Edge, the performance is much better than the GeoNet. So because for the GeoNet Edge, we are doing massive passing between the edges, which is able to better make use of the geometric information in the 3D structures. So that's the first part, which is supervised learning. Okay, and then in the second part, of course, we care about like pre-training. Can pre-train the model on labeled protein structure can first improve the performance. So here we just show one result. You can see that for this multiview contrast is based on the GeoNet Edge encoder. So you can see that So after pre-training, the performance can be further improved, showing that like pre-training can indeed help improve the model performance. So that's some experimental results. Okay. And then so here we also did some study on the sensitivity of pre-training on different data sets, because we can actually pre-train the models either on the PDB database, which the structures are obtained through experiments, right, which are more accurate. But then we can also pre-train the models on the structures predicted by AlphaFold 2, which of course is less accurate because it's obtained through prediction. But of course we have many structures because we can get as many as we want by running AlphaFold 2 on the protein sequence, right? So in general, we see that actually either pre-train the models on the PDB database or the AlphaFold predict database, we can get similar performance, showing that actually our pre-training methods is actually quite robust, either training on the real world experimental structures or the predicted structures. Okay, so that's an aberration study on pre-training. Okay. So this is the first type of like research we have been doing, which is really doing pre-training on 3D structures and then fine-tuning the models on some limited downstream supervised data set. Okay, and then we actually apply this methodology to a real-world application, which is called like antibody optimization. So antibody is kind of like biologics, right? So in drug discovery, usually, so let's say antibody drug development. So we usually got an initial antibody through animal immunization. So you do animal immunization and then you can get some antibody candidate. But usually the antibodies we got through animal immunizations, sometimes they are not ideal. For example, the binding affinity is not like strong enough. And then we need to do some optimization. We need to improve the binding affinity. Specifically here, if you look at this example, so we have a drug target called HEL, and then this is an antibody bind to this antigen. And then what we want to do here is, okay, we want to optimize this antibody to increase the binding affinity between the two. Okay, so how do we do this? So in reality, what we do is like we just want to make some mutations. We want to make some mutations on the interface between this antibody and this antigen. So we want to make some mutations. Most of the time, we make mutations on the CDR loops. We make like a few mutations to improve the binding affinity. Okay, so essentially, what we want to do here, what we want to do here is we actually want to build a machine learning model. We want to predict how the binding affinity changes if we make some mutations on the antibodies. So we want to build a predictive model on called like delta G, which is a binding affinity change. As we know, like for delta G is actually the difference between the binding affinity of the mutants and also the binding affinity of the wild type antibody. So that's the definition of our problem. Okay, and then for this problem, so we also actually use a similar approach. We also do like geometric pre-training and also fine tuning. Okay, so here we have like a pre-training model which is pre-trained on the 3D on labeled 3D protein structures, and then so we also have like a supervised learning model which is called a gear bind. For this gear bind model, so our goal is trying to predict the binding affinity change delta G. Okay, so once we have this like binding affinity change prediction model, okay, and then in reality, given an antigen and antibody complex, so we're gonna do in silico screening based on this binding affinity change model, right? Because we're gonna measure the binding affinity. We're gonna measure the binding affinity change for each possible mutation, right? And then we're gonna rank the mutations according to the binding affinity change. We're gonna keep the ones with optimal binding affinity improvement, right? And then that's how we can get some mutant candidates. Okay, and then once we do this, of course, this is still done in the in silico way, right? And then what we do is we're gonna further test those candidates in real world wet labs. Okay, so mainly like binding assays. So for example, we can measure like scores like ELISA or SPR. Okay, and that's how we can really got the true binding affinity change. Okay, so usually in the first round, we only like do some single mutations. Okay, and then after finish the first round of like experiments, and then you can see, okay, which single mutations is good. Okay, you can see the binding affinity change for different single mutations. So after the first round, and then we take the optimal single mutations, and then in the second round, you combine the best single mutations, and then you can first test the double mutations or the triple mutations, and that's how it works in practice. Okay, and there's a general framework of antibody optimization. So we can look at some details. Okay, so first supervised learning part. As I said, in this task, our goal is trying to build a machine learning model to predict the binding affinity change, which is essentially delta G, right? So for delta G, as we know, like is a difference between the binding affinity of the mutant and also the binding affinity of the wild type. Okay, so what we do here is like it's very similar to what we do before. Okay, so here's the structure of the wild type, which is antigen antibody complex structure, and then we're gonna construct a graph out of this complex. But remember that because here we only care about like binding affinity. So for binding affinity, it mainly depends on the interface between the two molecules. And that's why when we try to construct a graph, here the graph is only constructed based on the interface of the two molecules. Okay. And then, so we're gonna construct a graph based on the interface. And then once we have this graph, okay, remember this graph is not 2D. This graph is still like a 3D graph. So we're gonna do massive passing on the graph. So we can do different level of massive passing. So we can do residue level massive passing. We can do atom level massive passing. So we can also do massive passing between the edges, right? Which is able to make use of the angle information between the edges. Okay. So once we do the massive passing, so we can do pooling, and then we can get the repetition of this complex structure. And that's what we do for the wild type. So we can also do the same thing for the mutant, right? So we do the we construct the graph and also do the massive passing for the on the graph, and then we can get repetition for the mutant, and then we take these two as input to predict the final binding affinity change, which is delta G, right? And and this is our machine learning predictive model. So this is a trend based on some supervised label data. Okay, so this is supervised training. Okay, and as we just said, we're also gonna do like pre-training. Okay, so we also pre-train the model on some unlabeled protein structures. Okay, so here we actually also use a contrast learning idea. Okay, so here we have like a protein in a complex structure. Okay, and then so we also construct a graph based on the interface, and then we do geometric like deep learning. We can get repetition of this interface. Okay, so we do this for the wild type, and then we also do this like for the mutant. We can get a repetition for the mutant, and then for contrast learning, we just train a classifier to tell whether this is a true structure or a mutant, right? Okay, of course, you may have a question. In reality, how do you get the mutant? So what we do here is like, okay, given the real world protein complex structure, we just randomly mutate some residues on the interface. Okay, and then for each mutated residue, because you also need to determine the structure of the side chains, right? So how do we handle the structure of the side chains? So here we use a very simple approach. We just sample the side chain torsion angles from a Rotamer library. So it's not very accurate, but it's a quick way, and also some still a reasonable way to get the structures of the mutants. And that's how we calculate the repetition of the mutant structures. And then as we just said, we're gonna train a binary classifier or contrast learning to pre-train the 3D geometric encoder. Okay. And then so we do some in silico evaluations mainly on the ScanPy data set. So here we actually compare different kinds of methods. So the first two methods, FoldX and FlexDDG, so they are physics-based methods. So FlexDDG is based on the Rosetta energy function. Okay. And then the third method, bind DDG, is a deep learning method. Okay, and then our gear bind approach, and also the one with pre-training. So you can see that in general now on this task you can see that the deep learning method can significantly outperform the physics-based method, showing that nowadays, as long as we have enough data, so the knowledge we learn from the data can outperform the physics-based energy function. Okay, and then for our gear bind model, you can see after doing pre-training, so you can see that the performance further improves, showing that pre-training can indeed help the performance. And then we also do some ensemble. We actually ensemble all the physics-based methods and also the deep learning-based method. So you can see that after ensemble, so nowadays, like on this task, the Spearman correlation can achieve as high as 0.6. I think the accuracy is already good enough, can be used in practice. Okay, and that's why in reality, actually, we test the performance of our models on real world examples. So here, this one is a real world like antibody drug, which is called like CR3022. So this is like SARS-CoV-2 antibody, okay, which is obtained through from the patients. Okay, so we also know the complex between this and antibody and also the antigen. So here the antigen is the RBD domain of the SARS-CoV-2. Okay, so what we want to do here is like we want to make some mutations on the CDR loops of this antibody to improve the binding affinity. Okay, so we do some in silico screening to get which mutation can improve the binding affinity. So in the first round, we only identify single mutations because in our like label data set, we only have the binding affinity change labels for single mutations. That's why like for the predicted models, we only do single mutations. We don't have the label data for double or triple mutations. Okay. So in the first round, we pick like I think 8 optimal single mutations, and also we also take the best 2 single mutations as double like mutations. And then what we do is like we test all these like mutations or antibody sequence in the wet lab. Okay, so you can see that like in the first round, this one is wild type, this one is wild type. Okay, and the others are mutants. Okay, so in general, if the curve is like on the left top corner, the performance, the binding affinity is good. Okay, so in general, you can see that for most of the mutants predicted by our model, the binding affinity actually improves compared to the wild type. So this is in the first round. Okay, so in the first round, and then we can identify like a few good like single mutations, and then in the second round, we're gonna combine those like optimal single mutations. We're gonna test like double mutations and also triple mutations. Okay, so in the second round, eventually we found like molecules with 3 mutations, and the binding affinity improves like 17-fold compared to the wild type. So I want to emphasize, actually, this problem is already is very challenging because for the original molecule like CR3022, so the binding affinity is already very high. It's like 1 nanomole, and now we increase the binding affinity to like 6 to 10⁻11. So this is actually pretty challenging in practice. Okay. And then, so all these like experiments, like, so usually for every round of experiments, so this can be done in 1 week. So that's, so in total, we finish finish the computational and also wet lab experiments within 2 weeks. Okay, and that's pretty fast. You know, traditionally, how people tackle this problem. So what they do is like, okay, they're gonna like random mutate the residues, and then they're gonna build a library to do the screening. So traditionally, usually this take like 3 or 4 months, and now we actually do this did this like within two weeks, which is very efficient. Okay. And this is the first research questions, like how do we learn, how do we pre-train protein repetitions and also use those repetitions for different applications. Okay, let me see how much time I have. 10 minutes. Okay. For the second problem, we're gonna talk about like how do we like predict protein structures based on sequence. So we already have a lot of great work from DeepMind, like for example, AlphaFold 2, ESM4, and also the recent AlphaFold 3. Okay. So in our group, we have been also working on this problem. Okay. But you know, like for AlphaFold 2 and also AlphaFold 3, I think maybe many AlphaFold 2. So they mainly actually work on predicting the backbone structures. So they actually didn't do well on predicting the side chains of the proteins, and that's why we have been actually trying to working on building better machine learning models to predict the side chains of proteins. Okay. And side chain predictions is very important for problems like protein-protein interactions or protein-ligand interactions, because usually for two proteins, when they interact, they usually mainly interact through those side chains. Okay, and then for traditional approaches, we have methods like sampling from rotamer libraries, or you can do sampling from some physics-based energy functions, for example, like Rosetta 2. But those like physics-based energy functions, there are two limitations. First, they are not accurate, and also the sampling process is quite slow. Okay, and that's why we're also trying to define or develop diffusion models for sidechain prediction. For sidechain prediction, essentially we want to determine the coordinates, the structures of the sidechains of the proteins. And then, so one way to do this with diffusion models is essentially you can try to build the diffusion models on the Cartesians on the 3D coordinates of the atoms, right? And then in this work, we actually did it in a different way. So we actually didn't build diffusion models on the Cartesian space. We actually built the diffusion models in the torsion space, because you know, like for protein, for the side chains, so the degree of freedom of the side chains is not the coordinates of the atoms. The degree of freedom of the side chain is controlled by what? By the torsion angles, right? And that's why here we actually define a diffusion process in the torsion space instead of the Cartesian space. Okay. So specifically, we actually propose an autoregressive diffusion models. So you know, usually like for the side chains of proteins, we have like we have 4 torsion angles. So here we actually do this like in an autoregressive way. So we first like this is similar to what like ChatGPT is doing, right? So we first try to predict the first torsion angle, and once we know the first torsion angle, we condition on the first torsion angle, we predict the second torsion angle. We do this like autoregressively. Okay. Of course, you may ask, why don't we like predict all the 4 torsion angles simultaneously? We tried that. Actually, doesn't work that well, because so in reality, we found that actually the four torsion angles are very, very highly like interdependent. It makes the model very difficult to learn the dependency of the 4 torsion angles. That's why we found actually doing this like autoregressively make the learning process much easier. Okay. And then so we compare we evaluate the models on the CASP13 on the proteins in the CASP13 competition. Okay, so we compare our models with traditional like physics-based methods and also recent deep learning based methods. So our model can outperform all those like traditional physics-based methods and also the recent deep learning based methods, but with much like less parameters. Okay, I'm not going to get into details due to time limitation. Okay. And then so this is like on predicting the side chains of proteins. And this is another work we did recently, because you know, like for AlphaFold 2, one limitation is that for each protein sequence, we only predict one structure, and this is not true, right? Because in reality, actually, a protein can have multiple stable conformations, right? So essentially, so what we want to do here is, suppose this is the energy landscape of the protein structures. What we want to do is we actually want to sample. We actually want to sample from this energy landscape, right? So we actually want to get the structures with minimal energy. Okay, and that's what we want to do. 5 minutes. Okay, and then so there were a lot of like work in traditional biophysical domain. For example, people use like Monte Carlo monic dynamic methods to sample from either an energy function or a force field. But you know, for those methods, there are two limitations. The first limitation is of course, it's very computation expensive, and that's one big limitation, right? And the second one is usually most of the time they can be those like sampling method can be easily get trapped by some high energy modes, right? In other words, they cannot cover different modes of the energy function. Okay, and these are some traditional approaches. And recently, there are also some work. People have been trying to build a generative model from the molecular dynamic simulation data. So what people do is, okay, they first do some short run of molecular dynamics, and then they can get some training data, right? And then they fit a generative model to those like MD generated data, okay? And then there are also some limitations for this kind of approach. First, it's very expensive to get the MD simulation data, right? Because for every protein, so we have to do some rounds of MD simulation to get data, and then we will train a generative model, right? And remember that this kind of generative model is only specific to that protein. You cannot generalize this generative model to another protein. In other words, suppose you have a new protein, so you need to do this again, right? You need to do the MD simulation again, and you need to do train a generative model again. And that's the limitation of this kind of approach. So what we did here in this paper, this paper we published in this is iClear. So we train a generative structure translation model, not from the MD generated data. We actually train this model from the crystal structures from PDB essentially. Okay, so our approach is somehow actually inspired by traditional like approach called like a simulated annealing in MD simulation. So in those in the simulated annealing approach, so what did they do is like, so they have some like exploration phase. Okay, so you see the red one. Okay, so essentially what we do here is they are doing like exploration, and then locally they're gonna do some annealing. Essentially, they're gonna find the local minimal. They're gonna find the structure with local minimal energy, right? So here, what we do here is we're gonna start from a protein conformation. For example, the conformation can be predicted by AlphaFold2. Okay, and then once we have a starting point, we're gonna do exploration. So how do we do the exploration? So here we actually just do some stochastic perturbation. So we do some stochastic perturbation so we can cover different space of the energy landscape. Okay, and then once we do this, okay, we're gonna use our like generative model. Essentially, we train a score function. So we use that to map the perturbed structure to the local minimal, right? So we can do this traditionally using MD simulation, which is very slow. But now we do this using diffusion models, right? And that's what, because we have trained like such a structure to structure translation model, right? So we have like a noise, we have some noise structure, and then we try to map this noise structure to a stable structure with low energy. Okay, and this is based on the actually the IPA architecture in AlphaFold2. So I'm not going to go into the details. Okay, and then finally, so we've been working on like diffusion models for protein design. So I'm not going to the details because of time. And then so in my startup, besides working on the building the generative models or machine learning models, we also have built up our web labs so that like within like 2 or 3 months, 2 to 3 weeks, we can test like hundreds or even 1,000 proteins, and then we can get the labels of those proteins. We know, okay, what is the binding affinity, what is the functions of those proteins, and those label data is gonna be feedback to our machine learning models. We're gonna fine tune the generative models based on the label data and do another round of like design. Okay, so that's and that's very exciting. And so we are like designing different kinds of proteins like antibodies, enzymes, in different applications. Okay, so with that, I will stop here. Thank you.
Thanks a lot for the great talk. It's really interesting to learn about all the generative work that you've done for protein and representation work, and also to see that it works in real wet lab experiments. So that's I think it's a good sign that like all the work that everyone here is doing like will have an impact in drug discovery. We have time like 2 minutes for questions. So for each talk lasts roughly 40 minutes. And we have like a few minutes in between the talks only for 1 or 2 questions. For the questions, like whenever you have them, you can line up at the microphone. But usually we rather keep them after the talk. So go ahead.
Hi, so it's very interesting. The protein problem is very interesting because you have to make a choice about resolution, what you're going to model as the nodes of the graph. So for example, you could have had higher resolution, the nodes of the graph could have been atoms instead of residues.
Yeah.
You could have had lower resolution, you could have modeled an alpha helix as a rod.
Yeah.
So how do you make that decision?
Yeah, that's a good question. I think ideally you probably want to ideally you want to represent the proteins at the atom level, right? That's ideally okay. But usually there are a few challenges because if you represent proteins as atom level, then your graph is going to be huge, right? So because then you probably have like 1,000 atoms in a graph, and that's very computation expensive, and that's one thing. And the second one is like if you represent proteins as atom graph, it's going to be very sensitive because sometimes like you may not be able to get a very accurate protein structure, right? If you have an experimental structure, that's fine. But sometimes like even the experimental structure is not accurate, right? Most of the time you can only get the structure by running AlphaFold tool, right? Which the side chains are not accurate. So I think so there are some limitations on as I said, ideally atom graph is optimal, right? But there are some challenges. But in some cases, I think atom level is actually quite helpful. If you see in our case, when we try to model protein-protein interaction, we actually don't care about the entire protein-protein structure. We only care about the interface. So in that case, we can only take the interface part, and then the graph is very small, right? Okay, and that's one thing. Okay. And then, so in reality, I think it's good. Like you combine different resolutions, atom level and the residue level. So residue level, yeah, is the good thing is that it's less accurate, but it's more robust, right? Yeah. So, and that's what we found. You always like combine the two. It's always ideal. Yeah.
Great, thank you.
Thank you for the great talk. I might have missed this, but like can you hear me? I might have missed this, but like in your second part of your talk, when you present the string to string method, like how did
So you mean
how did you address the sidechain conformational sampling problem? I might have.
Right. So I think we actually do an approximation. We first sample the backbone structure, and then given the backbone structure, we actually use some Rotema library. It's not 100. It's not that accurate actually. So we actually do some approximation there. So we actually mainly train a general model to sample the protein structures and the backbone structures. And once we have sampled the backbone structure, we condition the backbone structures. We use some Rotema libraries to quickly to sample the side chains. It's not that accurate, but it's a relatively efficient way and accurate way. Yeah.
What's the Rotema library you mentioned?
So Rotema library is some kind of like so people for different torsion angle for each torsion angle there's some distribution. It's not totally like a random. So people got that statistics from the PDB database. So basically, for every torsion angles, you have some statistics from the PDB database. And then what people do is you're gonna draw a sample from that distribution. That distribution is got from the PDB database.
Okay.
That maybe is a intuitive like explanation. Yeah.
Thank you.
Yeah.
Right.
Hi.
Let's make this the last question.
So my question is about the contrastive loss you're using for the 3D geometric and structure encoder.
Yeah.
So you mentioned that the positive pairs are structural motifs from the same protein.
Yes.
And the negative pairs are the one from different proteins.
Yeah.
But I wonder like how you make your training set then, because sometimes you may have like proteins that are very structurally similar in the same, like,
Yeah.
yeah. So what if you pick motifs from one structure, but then the negative would be like a protein with very similar?
Right. So I think there's always a problem like this, like in contrast learning, even in the case of like image repetition, right? So you take two patches from the same image, they may not be similar, right? And the two patches from different image, they may be similar, right? So, but I think this is more like a signal versus noise ratio. It's more likely to happen, right? Patches or motifs from the same proteins, they are more likely to be similar than the ones from different proteins. So I think it's more like from the statistical point of view, or signal versus noise ratio.
Okay.
On some specific cases, it doesn't hold for sure. Yeah, but the good thing is that for machine learning models, we can learn from the noise data.